Thanks for joining us on another exciting journey into the world of science, right here on the Sand Science Experience. If you enjoy this kind of content, hit like and subscribe. It's the best way to support the channel and keep these episodes coming. Welcome to the Deep Dive. Today, we're not just scratching the surface. We're going deep, really deep, into how protein turnover changes as organisms get older. And our guide for this journey, it's the tiny nematode worm, Caenorhabditis elegans.
Yeah.
C. elegans.
That's right. And protein turnover, just to recap quickly, is this constant cycle, building new proteins, breaking down the old or damaged ones. It's like cellular inventory management.
Constantly refreshing the stock.
Exactly. And it's absolutely fundamental. So understanding how this changes, even in a simple worm, can give us some really profound insights into aging itself, and maybe even ways to intervene down the line.
Absolutely. And we're grounding this discussion in some solid research. We've got a scientific paper here titled Changes of Protein Turnover in Aging Caenorhabditis elegans. So our mission today is basically to unpack the key findings. How does this protein balancing act shift as C. elegans ages, and what might that mean for, the bigger picture of aging?
Let's do it.
Okay, so kicking things off, what's the headline? The main discovery about protein turnover as these worms get on a bit.
Well, the main takeaway, the sort of big picture, is that, yes, protein turnover generally slows down as C. elegans gets older. This isn't hugely surprising. It actually fits with what's been seen in other organisms.
Right.
The study specifically found that the median protein half-life, that's, how long it takes for half of a particular protein pool to be replaced, it basically doubles. It goes from day 1 of adulthood to day 7.
Wow, doubles. So the cells are definitely taking longer to refresh their protein components as the worm ages. But you mentioned generally it's not quite that simple, is it? It wasn't a uniform slowdown across the board.
No, not at all. And that's where it gets really interesting. While there's this overall trend, they found that only about 40% of the proteome, all the proteins in the worm, actually showed this kind of gradual slowdown.
Only 40%. So what about the other 60%?
Well, they didn't follow that neat pattern. And perhaps even more surprisingly, a very small fraction, only about 7%, actually showed increased turnover rates with age.
Increased.
Okay.
That definitely goes against the simple idea of everything just winding down.
Yeah.
So why the difference? Why isn't it all slowing down together?
The researchers called it heterogeneous. Think of it like different departments in a factory. As the factory ages, maybe one production line slows down significantly, another keeps pace reasonably well, and maybe one quality control section has to work even harder.
Okay, that makes sense. Different parts aging differently.
Exactly. When they looked at functional groups of proteins that work together, they didn't see consistent patterns within those groups either. It suggests that the decline in managing the proteome isn't this, centrally controlled, coordinated shutdown.
Right, so it's less like a master switch being turned down slowly.
Yeah.
And more like lots of individual switches and circuits behaving kind of differently as time passes.
That's a great analogy, yes. And amidst the sort of mixed picture of slowdown and variability, there was one area of cell maintenance that seemed to be, holding up quite well.
Ah, the cellular housekeeping. Tell us about that. How does the cell try and keep things clean and functional even as it's aging?
So this was quite striking. The turnover of proteins within the ubiquitin-proteasome system, the UPS, which is basically the cell's main machinery for tagging and degrading unwanted proteins.
The recycling center.
Pretty much, yeah. That system's own components seem to maintain their turnover rates quite well in the older worms.
Okay, so the machinery for getting rid of old proteins is staying relatively refreshed itself. That sounds important.
It seems very important. And similarly, antioxidant systems, the ones fighting off, oxidative stress, damage from reactive molecules, those proteins also maintain relatively fast turnover rates. So the idea is that maintaining these quality control and defense mechanisms might be a key protective strategy. The cell is trying to keep its repair and disposal crews in good shape.
Like prioritizing the fire department and the waste management in an aging city.
Exactly. It seems like a sensible strategy. But there's always a but, isn't there?
Yeah.
The study also notes that despite these efforts, keeping the quality control running, the widespread protein damage and eventual collapse of the proteome still seems to happen in the end.
So it helps. It potentially delays things, but it doesn't ultimately prevent the age-related decline.
That seems to be the case. Even the best maintenance crews can't keep an old structure standing forever, perhaps.
Okay. So we've talked about protein breakdown. What about the other side of the coin? The machinery that builds the new proteins. How did that look?
Well, that's where the picture looks a bit different. When they looked at the protein synthesis machinery, so ribosomes, translation factors, all the components needed to actually build proteins.
The protein factories.
Yeah. They saw what they describe as a fanning out of the half-lives of these components as the worms aged.
Fanning out. What does that mean practically?
It means increased variability. So imagine when the machinery is young, all the parts are working at roughly the same efficient rate. Their replacement rates are similar.
Oh.
But as the worms age, some components start getting replaced much, much slower, while others might change less, or maybe even faster in some rare cases. The range of turnover rates within that system widens considerably.
So the coordination breaks down. The factory parts are aging at different speeds, making the whole production line less reliable, maybe.
That's a good way to think about it. Increased heterogeneity within the system itself. And another piece here is that they also found that a significant number of these protein synthesis components tend to clump together, aggregate with age.
Protein aggregates. That sounds bad for function.
It generally is, yes. These clumps can physically get in the way and stop the machinery from working properly.
Okay, so we have this complex picture. Some things slowing down, quality control, trying to keep up. But the factories making new stuff are becoming less coordinated and gummed up. Did they look at any specific protein types to give us concrete examples?
They did, yeah. And looking at specific groups is really helpful. Take tubulins and vitellogenins, for example.
Okay, tubulins are structural proteins, right? Like cell scaffolding.
Exactly, part of the cytoskeleton, crucial for transport inside the cell. And vitellogenins are yolk proteins, nutrient reserves.
Different functions. What happened to their turnover?
Both groups showed a really consistent, quite strong slowdown in turnover as the worms got older. Very clear trend for both.
Interesting. Same trend, different proteins. Did they behave similarly in other ways, like with that aggregation problem?
No, and this is a key point. Tubulins, despite their slowing turnover, didn't really show much tendency to aggregate. But vitellogenins did aggregate significantly.
Okay, so the fact that tubulins slow down without aggregating suggests that aggregation isn't the only reason turnover slows down.
Precisely. It decouples those two things, which is important. Something else is causing or allowing tubulin turnover to slow, independent of clumping.
And what might be the consequences? Slower tubulin turnover could mess with internal cell transport.
That's definitely a possibility. It could contribute to a general decline in cellular logistics with age. And for vitellogenins, the combination of slower turnover and aggregation means they really build up, which might cause its own set of problems even in worms not actively making eggs.
Right. Okay, what about muscle? Muscle decline, sarcopenia, is a big feature of aging for us. How does it look in the worm?
Very relevant question. The study found that muscle proteins are actually incredibly stable. They showed very, very little turnover throughout the worm's life, even when young.
So they're built to last, basically.
They seem to be. But that lack of turnover, that stability, might actually become a liability with age. If muscle proteins get damaged over time and aren't efficiently replaced because their baseline turnover is so low, that could directly contribute to the decline in muscle function.
So it's not necessarily about turnover slowing down with age for muscle, but maybe about it being too slow to begin with to handle accumulated damage over a lifetime.
That seems to be the implication, yes. It really highlights how complex this is. Sometimes slow turnover is bad, sometimes maybe a lack of turnover is bad.
Okay. Any other tissues stand out?
Well, contrasting with muscle, they found that the pharynx, the worm's feeding organ, its throat basically had a higher proportion of proteins with increased or maintained fast turnover rates compared to muscle.
The pharynx.
Yeah.
It's interacting with the environment more directly, right? Food, microbes.
Exactly. The thinking is that because it's on the front lines, facing more potential stress and damage, it might need a more dynamic protein replacement system to cope. Faster repairs, faster replacement of damaged goods.
That makes a lot of sense. What about the engine room? Energy metabolism. Surely that changes with age.
You'd certainly expect it to. But again, no single simple pattern emerged for all energy metabolism proteins. It was a bit of a mixed bag.
Any specific trends within that?
There were some hints. They noted that some enzymes involved in glycolysis and the TCA cycle core energy pathways did tend to slow down their turnover. But interestingly, proteins involved in fatty acid metabolism, breaking down fats, seem to have unchanged or even slightly increased turnover.
Okay, so maybe a shift. Less reliance on sugar pathways, maybe more on fats as the worms age.
It could suggest something like that, yeah. A metabolic adaptation in older worms. It's speculative, but the data points that way. Needs more work to confirm, of course.
And antioxidants. You mentioned they kept up turnover earlier. Any specific examples?
Yes, enzymes like SOD1, a key antioxidant enzyme, showed increased turnover with age.
So the cell is actively making more or replacing that defense enzyme faster as it gets older.
It seems that way. It looks like an active response to the increased oxidative stress that's a hallmark of aging. So it's not just passive decline. There are active countermeasures happening at the protein level.
Fascinating. Okay, so we have turnover rates changing all over the place. How does this relate to the actual amount of each protein present? Does slower turnover always mean more protein and faster turnover mean less?
Great question. And the answer is, it's complicated. They looked at this relationship between turnover changes and abundance changes, and it wasn't a simple one-to-one correlation.
Give us an example.
Okay, ribosomal proteins, the ones involved in making proteins. Their overall abundance tends to decrease with age. But, as we said, their turnover rates were all over the map, some slowed down, some sped up.
So lower levels overall, but turnover changes didn't consistently predict that decrease.
Exactly. It shows that the amount of protein you have is a balance, right? It depends on both how fast you're making it, synthesis, and how fast you're breaking it down, degradation. Changes in either one or both affect the final level.
So you can't just look at turnover and predict abundance or vice versa.
Not always. Contrast that with vitellogenins. Their turnover decreased and their abundance increased. In that case, it seems the slower breakdown is likely a major reason they accumulate. But for tubulins, turnover slowed, yet their abundance didn't change much, suggesting maybe their synthesis rate also decreased to compensate.
Wow. Okay, it really is an intricate dance between making and breaking proteins.
Yeah.
So let's try and pull this all together. As we wrap up this deep dive on C. elegans protein turnover and aging, what's the main message?
I think the core message is the complexity and the heterogeneity. Aging, at least in terms of protein management in these worms, isn't a simple uniform process of decline. It's messy.
Messy, okay.
Yeah, you have this general trend towards slower turnover for many proteins, but significant variability. You have critical quality control systems like the UPS and antioxidants that seem to be actively maintained, perhaps protectively.
Right.
While other crucial systems, especially protein synthesis, show signs of dysregulation, more errors, more variability.
So it's a patchwork of changes, not a blanket slowdown.
Precisely. And understanding this patchwork, this heterogeneity, is probably key to understanding the aging process itself.
In studying this in a tiny worm, C. elegans, gives us fundamental clues that could apply more broadly.
Absolutely. These are basic cellular processes. The details will differ, of course, but the fundamental challenges of maintaining a functional proteome over time are likely shared across many species, including us. This research provides really valuable molecular insights.
It really does. And it leaves you thinking, if maintaining that quality control machinery, the protein degradation and antioxidant systems, is so important and seems to be prioritized by the aging worm, well, what strategies could we potentially explore to maybe, boost those systems ourselves? Could we enhance that natural maintenance?
That's the million-dollar question, isn't it? It's certainly a major focus of aging research, how to support cellular quality control.
Definitely food for thought. This has been incredibly insightful. Thanks for breaking down that complex paper for us.
My pleasure. It's a fascinating area, always evolving.
Thanks for listening. And as always, don't forget to like the video and subscribe if you haven't already.